My name is Kazuki Hiro. I'm glad to participate in this conference. Now I'd like to present my study titled An Application of a Berthelot Method Using a Metal Tube to Inactivation of Bacillus subtilis. I would like to present my study in this order. In introduction, I will explain a liquid under negative pressure and so on. In experiment, I will explain a Berthelot method and so on. In result and discussion, I will explain negative pressures for bacterial solution and so on. And lastly, I will conclude my study. At first, I would like to introduce a liquid under negative pressure. A liquid is enclosed in a cylinder with a piston. As the piston is moved out, the liquid pressure decreases and can become negative. But if air bubbles penetrate into the cylinder, negative pressure does not build up. Therefore, a liquid under negative pressure has been difficult to be generated. However, negative pressures in liquids are important and interesting objects in science and technology. For example, negative pressures may inactive bacteria. This graph shows a stability diagram of Escherichia coli. The diagram seems to be partly elliptical in a positive pressure temperature plane. The bacterial count decreases by 2 orders of magnitude within 5 minutes out of the elliptical region. If the elliptical bottom seems to be in a negative pressure region, which is much more accessible than a positive one. To author's knowledge, there have been no studies on inactivation of bacteria in negative pressure regions. Thus, the purpose of this study is as follows. In order to investigate inactivation effects of negative pressures, a solution including a kind of bacteria is exposed to negative pressures. A number of bacterial colonies is counted by an agar dilute plate method. Now I'd like to explain experiment. There are few experimental methods to generate negative pressures. A Berthelot method using a metal tube is the most feasible because it can generate static measured negative pressure for sample liquids of not too small volumes. I would like to explain a Berthelot method using a metal tube. This figure shows a liquid sealed in a metal tube, and this graph shows a relation between pressures and temperatures schematically. When a tube is heated, the liquid pressure increases because of a thermal expansion coefficient of the liquid higher than that of the metal. On the PT graph, the pressure increases to the point A like this. Next, when the tube is cooled, the pressure decreases for the same reason. On the graph, the pressure decreases to the B like this. And further cooling, negative pressure builds up. On the graph, the pressure decreases like this. By further cooling, if tiny bubbles exist in the system, cavitation occurs suddenly. On the graph, cavitation occurs at the C, and the pressure jumps sharply to the D. At the D, the liquid coexists with its vapor, like this. Next, as the tube is heated again, the vapor disappears gradually in the liquid. As the tube heated further, the pressure begins to increase steeply. This cycle A, B, C, D, B, A. This cycle A, B, C, D, B, A is called a temperature cycle. A repetition of temperature cycles make negative pressures high. This figure shows a metal vessel tube. The left side shows the overview and the right side shows the inside view. The tube consists of 5 main parts, namely a screw, a socket, a nut, a ball, and a pressure transducer. The specimen chamber has a volume of circa 15 cubic millimeters. This figure shows experimental apparatus. The tube is immersed in the hot bath and suspended in the air alternately by motor control. The liquid pressure is positive in the hot bath and negative in the air. The bacteria in the solution is Bacillus subtilis subspecies. The solution, including 2.5% neutronic broth as shown in this table, is tested as a sample liquid. In order to evaluate an inactivation effect, a definite number of temperature cycles are carried out, and then the sample solution is extracted with a micropipette carefully, and the extracted solution is cultured by an agar dissolution plate method for 2 days at 37 degrees centigrade in an incubator, and a number of colonies is counted and is compared with those which did not undergo temperature cycles. Next, I would like to present results and discussion. This graph shows a typical relation between negative pressure and temperature cycle for a bacterial solution. Negative pressures from circa 1.5 MPa to circa 6.5 MPa in magnitude were measured. This table shows average and standard deviation for definite cycles. Irrespective of temperature cycles, negative pressures of circa 4.5 plus minus 1.5 MPa were measured. These photographs show typical result of an agar dilution method. Figure A shows the case of no cycle and the B shows the case of 15 cycles. The number of colonies of 15 cycles were less than those of no cycles. This graph shows a relation between reduction rates and cycles. A reduction rate is defined by this equation, where N0 represents colony count for no cycle and NC represents colony count for constant cycles. Reduction rates increases with cycles. A possibility of the metal vessel method as a new means for an inactivation of bacteria was demonstrated. Next, I would like to conclude my study as follows. In order to check inactivation effects of negative pressures on bacteria, solutions of Bacillus subtilis underwent negative pressure repeatedly using a metal vessel tube. An agar dilute plate method indicates that numbers of colonies which underwent negative pressures were less than those of with no cycle, and reduction rate of bacteria increased with cycles. A possibility of the metal vessel method as a new means for an inactivation of bacteria without any chemical compound was demonstrated. Thank you very much.